Alright, hello everyone. This is going to be our first video for the semester. There will be a brief review on scientific method. Now, some of the words we'll use are similar. Some of the words from whatever whatever middle school you're coming from might be a little bit different, but these are the words at Marist you're going to use throughout. Okay, so make sure to take notes, follow along. Like I said, I post these lectures online. And then you can just print out and put in a PDF of the PowerPoint. That way it makes it easy to follow along and take notes. All right, so scientific method. So it's just a systematic way of solving problems. So you have a problem. Really, you can use scientific method for a lot of things in life, not just necessarily science. But so a few parts, keys to the scientific method. There are 7 steps. Sometimes you'll see 6. It depends on which one where you're coming from, things like that. But these are the 7 that we're going to do. First, you're going to state the problem. Sometimes you'll see this as also observations, and then you'll come in and you'll state the problem. So what's going on? This is the old if then statement. Now, it's not my favorite way to do it. I like a little bit deeper than if blah blah blah, then blah blah blah. But especially at the beginning of the course, it's perfectly fine. So number 2, we're going to do the research. You're going to have your hypothesis, which is, like I said, kind of your if-then statement. Your experiment, the data collection, your conclusion, and then one that you may or may not have been acquainted with yet is repeat the work. All right? Just because I do it once and it works doesn't mean that necessarily it's going to work again. So if we're doing for labs and things like that, we usually don't have time to do multiple experiments, multiple tests. But if we were doing research in a lab, we would do each test or whatever we're doing multiple times to make sure that the results we got are verified. All right, so let's go through all 7. All right, so step 1, we're going to state whatever that problem is. In this case, how long does it take how long does it take for cereal to get soggy in the milk? Okay, so like I said, this isn't the most scientific, super in-depth question, but it's just a good question for an example. Next, we're going to do our research. Here, we're going to gather information on our topic. Okay, now information can come from books, a computer, or an expert. This would be somebody who has done research in the topic. Now, in the field of cereal sogginess, there's not a lot of experts, but in other things that you may or may not want to research, there are experts. There's people who have devoted their entire life to different aspects of parts of science. Step 3, form a hypothesis. All right, so this is where we're going to make what we call an educated guess. Now, when I say educated guess, I don't mean that we're just going to haphazardly make a hypothesis. All right, we did our research in the last step, so now we have an idea of what should happen. Now, if your hypothesis is wrong, what do you think Mr. Rogers is going to give you for a grade? All right, it doesn't matter. I don't care if your hypothesis is wrong. Okay, the experimenting is not about my hypothesis has to be right. The experimenting is about finding out your answer. So even if your experiment is, or even if your hypothesis is wrong, you have learned something from it. So do not worry about, oh, my hypothesis is wrong, I got to change my answer, I got to go back and, no, do not do that. You report the data, you give it as is. It does not matter. You will never get points off for having a wrong hypothesis. Okay. A lot of times we'll put them in if-then. We'll talk about more advanced ways to do that, but for now, if-then is perfectly fine. So an example, if I add 1 cup of milk to 2 cups of cereal, then the cereal will be soggy in 5 minutes. So that the key here with a hypothesis is that it is testable. Okay? If it's not testable, it's not a valid hypothesis. I have to be able to come up with an answer out of it. So I can't even think of a non-testable hypothesis off the top of my head because I've been doing this for so many years. But it has to be testable. If it's not testable, then it's not valid. If it's not valid, you don't get points for it. Perform your experiment. All right. So just a test to show whether your hypothesis is true or not, or accurate or inaccurate. So a few things as we go over this. So variables. In our experiment, we have variables. These are things, there's a host of variables. There's controls, there's experimental, there's all kinds of things. Okay. So variables are just factors in the experiment. Okay. You have your control variable. These are the ones that remain the same. So like same bowl size, same amount of cereal. Let's say same bowl size, same amount of cereal. We'll use 100 grams. All right. And we'll use, we'll use 200 grams of milk. So these are going to be kept constant. Really the only thing that's going to be changed is our independent variable. I'm going to put this in parentheses. Sometimes in what we used to hear, sometimes in middle school, is the manipulated variable. This is the one that has changed. This is the only one that has changed. Everything else is held constant except for this one. So we use like cornflakes. Lucky Charms. Other cereals. All right, I don't need a lot of cereals, so I'm having trouble with examples. But we're just using different types of cereals. This is going to be our independent variable. Now our dependent, our responding variable, is what we are getting or what you are measuring. Okay. And we'll talk about this a little bit in the next video, but it never hurts to go in and drop some knowledge down here. This is always going to be your x-axis. This is always going to be your y-axis when you're graphing. We'll talk more about that in the graphing video. But just to know, your independent, or what I change, goes on the horizontal. All right. And what the outcome is goes on the vertical. So experimental variables, we have control. So here we got our bowl size is the same. So bowl, milk, can't write this morning. Independent variable, types of cereal. All right. So dependent is, what's our dependent variable here? How long it takes to get soggy. Okay. So step 5, record, and we will analyze the data. Now, data is just information that we get from the experiment, right? That's it. We have two main types of data that we talk about in science. We have qualitative and quantitative. So quantitative right here and qualitative right here. So I'm going to talk over qualitative first. Qualitative has the word in there. You can see it's like quality, okay? So these are things such as color, texture, smell, taste, etc. Like blue, for example. All right. At Marist, you have a light blue polo. The seniors have a dark blue polo. You have variations of blue on the for like different sports teams and things like that. So if I said the color of his shirt is blue, it's like, well, which blue are you talking about? There's a whole bunch. Okay. So this qualitative is not bad data, but it is not necessarily what we're always looking for in science, because it can be kind of argued or debated. Like the texture. What is smooth or rough to one person may be smooth or rough to somebody else. Okay. Smell. Oh, that smells good. Nope, that smells disgusting. All right. Those are different. Taste, all right? We'll have different taste buds and different smellers, okay? Olfactory senses. So a taste might be something different to me than what it would be to you. Now, quantitative, we have that word in there kind of like quantity, right? So this is what we usually look for in science is quantitative data. Now, we can't always take quantitative data. Some things, especially if you're doing like behavioral ecology studies and things like that, don't necessarily lend itself to quantitative data. Sometimes we have to take qualitative. But for most of our labs, we'll take quantitative data. These are measurements. These have numbers and not hashtags. That's the number symbol. Temperature, height, weight, well, more or less mass. Should be mass, not weight. Mass, things of this nature. Like if I said there are 7 children in the classroom, that is a fact or not. It is yes or no. But there is one type of blue in the classroom. That is definitely not happening, right? There are multiple types of blue in every classroom you will be in this year. So the data is always represented in a table, pictures, graphs, something, okay? A lot of times we don't necessarily post or put our data table down. If people ask to see it, that's fine. But we usually post the results, the graph, the picture, the whatever we took to kind of surmise the data as opposed to posting this massive, long data table. All right, next we come to a conclusion. This is where we state that our hypothesis was accepted or rejected, meaning that our hypothesis is right, so we accept it, we reject it if it was wrong, and it answers your question. So back to the question, it was how long will it take for cereal to get soggy? All right, so we do a couple tests. We find out that I said, well, we said, I think in the example, it's 5 minutes. Well, actually, it's like 10 minutes. So in this case, if that was the case, we would reject our hypothesis. And we would just talk about that kind of stuff in our conclusion, why we rejected our hypothesis. Then last but not least, retest. Do the experiment again, all right? I don't know about you, but usually the first time I try something or do something, I mess it up at least a little bit, okay? So we're probably going to mess something up, and that's okay. So we want to go back and see if our results are congruent, similar, very similar to our original test. And if they are, then we can say, yes, this is a valid test. If not, then we have to go back and figure out what we did wrong. So that's it for this video. If you have any questions, comments, or concerns, come on in to tutorial and we will get them straightened out.